In this video, we will discuss about bacteriophage therapy. So before proceeding directly into the topic of this bacteriophage therapy, let us first quickly understand why people are focusing on bacteriophage therapy. So because before the present approach, that is antibiotic approach to treat various diseases, is becoming inefficient with time because most of the bacteria or infection get resistance against the antibiotic with time because of the uh, natural evolution, and because of that, and this leads to the inefficiency of the antibiotics that are currently being used. So there are two approaches left with them: either to continue finding out new molecules that can target against these bacteria that are causing infection, or to focus on completely new approach, that is bacteriophage therapy. And this approach will not have that much disadvantage that antibiotic approaches has in terms of making uh, the bacteria resistance to the antibiotics. As and another advantage of this bacteriophage therapy is that, see, because its natural target is bacteria, it can directly infect the host, or we can say bacteria. So now let us understand how this bacteriophage therapy works. So this is the bacteriophage that contains the gene of interest, or we can say the gene that can kill the cells that are infected with bacteria. So the first step is the binding of this bacteriophage to the cell surface or bacteria cells. After that, the it will inject the gene of interest into the inside the cell of the host, and then the there can be two steps of there can be two steps by which the replication of this uh, injected gene occurs. First one is the uh, lysogenic phase, in which the integration of the DNA is done uh, to the bacteria phase DNA, to the host DNA, we can say. Integration of the injected DNA is done with the host DNA, and in this case, the gene transcription and translation occurs along with the host DNA, as we can see here. And the another approach that can happen is that the transcription and translation of this gene of interest will happen autonomously, or we can say independent of the transcription and translation of the gene of uh, the host chromosome. So in both the cases, the final result is that the replication of viral genome and synthesis of viral protein increases. And as this replication of viral genome and synthesis of viral particle increases, what happens is that the more bacteriophage will produce within the cells, and they will be transported outside the more bacteriophage will produce within the cells. And at the same time, the uh, function of this gene of interest, or we can say the gene that is injected through bacteriophage, will do its function to kill the cells kills or the cells, or uh, signals the cells towards programmed cell death. And when the cells lysis got degraded, the bacteriophage can spread, or it will be released and spread across uh, different parts of the cells that are infected with bacteria, as it is natural as its natural ability is to bind to the bacteria. So it is there are very much possibilities that it will bind to the cells that are infected with the bacteria. Well, more this therapy is as this therapy is at its initial stage and needs to be further developed before it is commercially available or before it is commercially applied to the patients. So if you need to understand more about this bacteriophage therapy, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.